I think recently there's been a number of developments which are exciting. Obviously the increasing capacity of something like the Illumina NovaSeq is very exciting, but what we really need for whole genome sequencing in order to be able to apply that in a clinical context, which is what I'm interested in, is as an increase in throughput and a decrease in price. And so it's encouraging that something like the BGI-seq is coming along, which might perhaps offer an alternative and maybe reduce prices or at least introduce some competition into the market. And then of course there's the possibilities of long-read sequencing, which has a number of great possibilities for structural variant detection. So I think there are some very exciting developments in the last year. Right, so in my lab, which is a clinical genomics lab, and we're trying to take some of the latest and most interesting methodologies and move them across into the clinic. And in order to do that, the key driving factor for us is high throughput. It's high throughput and quality. So we need to be able to take hundreds of samples and put those across the same assays at the same time. In order to do that, the logistics of that mean automation, lots of automation, multiple indexing, so that we can take the biggest benefit, if you like, from the technologies that are out there, but also quality control. So all of this stuff has to be done within the context, within the envelope of a very high quality workflow. So all of those things coming together, I think, are going to enable a better clinical application of the sequencing technologies. I think high throughput sequencing, so we know that Genomics England and NHS England now are going very big on whole genome sequencing for clinical diagnostics. I still personally think that's a couple of years away for proper application. So the high throughput sequencing methodologies will also be complemented by some other methodologies like droplet digital PCR and perhaps some of the long read technologies for looking for gene fusions, like the nanopore technology, which I think has got great potential for gene fusion detection. Well, I hope it's going to be reduction in costs and reduction in prices. I think that's probably what everybody else would also want. There are multiple other technologies out there, but of course we're all well aware that there's one kind of big beast provider, and they do have somewhat of a domination on the market. I'm hoping that there'll be new advantages that will allow us to reduce those costs. In terms of new technologies that are coming along, I think the maturity of the long read technologies is going to offer us things that we're not able to do currently, and I think that's very exciting too.